Welcome back friends, welcome to another lecture from Just Biology. In this lecture we're going to talk about termination process in prokaryotic cells. In the last lecture we have talked about elongation process in prokaryotic cells. In this lecture we will be focusing on the termination process. We know that the DNA of the prokaryotic cell, so they have a different, this is the replication bubble and this is also. So this will be joining some point. So until the DNA polymerization we can see. So where actually the point meets each other. So DNA is supposed to be synthesized at that point. So this is the scenario. So this is the replication bubble and this is the replication bubble. And now so actually the fork, if you see the fork of this replication, this is the one fork, this is second fork. So we can term it like RF1 and RF2. So if you see this RF2, so it can move only this direction. So we can call it as clockwise or parallel direction. We can call it as, like in this direction, we can call it as clockwise direction. And obviously this direction we can call it as anti-clockwise. So we can call it as clockwise and anti-clockwise. So these two fork will be meet at a particular point. Let's say this is the point where they meet together. So this point we can call it as termination site or T site. So termination site. So if you see this termination site, we can see a specific element or the specific sequences which is present in the termination site. So they are often made up of 24 base pairs long. So they are the 24 base pairs length. So you can see, so there are different kinds of termination site which is present in each and various prokaryotes. So not only in E. coli, we can see if you take any bacilli or staphylococci or mycobacterium, so you can see a different combination of the termination site. So actually the termination site is made, actually they are 24 bases long and that varies from A to J. So they have the termination site that we can call it as TER site. So TER site, TER A, TER B, TER C and so on till J we can see. Now the question is, if the circular DNA is possessing a one replication bubble, so that's quite fine. So that is fine also. But in some case they need to form more than one bubble. So where the problem comes in. So where we can see the problem. So I'll tell you how they are managing all those steps in while when they formed more than one replication bubble. So let's erase this picture and I'll draw it again for the better understanding. So this can be one bubble and this can be another bubble. So that meets together at the same point. So here you can see hydrogen bond. Here also you can see the hydrogen bond. This is a broken hydrogen bond, and here also you can see that. So this is the bubble. So this is the scenario. This is a replication bubble RB1 and RB2. So this is the two bubble which they formed. Now the question is, so I told you they have a specific site is called as termination site. So we can call it TER site. We can call it as TER site. So I have told you they are made up of 24 base long. 24 base long and they are often A to J. So the TER A, TER B, TER C and so on till J. Now the question is, so let's say so this is So one of the termination side here, and here also we can see one of the sides here. So we can term this like, so this is TER A and this is TER C. Let's imagine. So we have the two sides. We have got the sides TER A and TER C. Now this bubble RB2 will move a clockwise direction and RB1 will go anti-clockwise direction, and that we know actually. Now how this process will be blocked and how this termination is actually catalyzed. So there is a protein which catalyzes this termination process. So that protein we can call it as TUS protein. We can call it as TUS protein. So this TUS protein will have a specific architecture. The TUS protein will have a specific architecture. So it has got two kinds of ends. We can call it as permissive end as well as the non-permissive end. So better I'll draw here. So this is so I have to draw a little space apart because this is quite near. So I have to draw it a little far away. So let's write it here. So this is TER A. And this is TER C. Now, so I told you there is a protein is called termination utilizing sequence protein. So it will bind with this kind of sequence. So this is the sequence. So they have it has got bound with TER A site, and same way here also. So now they formed a TUS-TER complex. That means a termination utilizing protein that is bound with termination site. So that is TER A and TER C. So they formed TUS-TER complex. So this makes sense here. This TUS-TER complex, what it is? Actually, this is going to be stop the movement of the DNA B protein, or they will not allow this protein to do the polymerization further. So that's why this complex makes sense here. If you see the structure of this TUS protein, they have two different regions. They have got two ends actually. So I would say the end. So this, let's say this is the protein. So it has got, I told you it has got two ends. So this end we can call it as non-permissive end. So that means the DNA B protein cannot pass through the non-permissive end. So that is the idea. So this is permissive end. So in this permissive end will allow this B protein to pass through it. So this is the two end which is present in the TUS protein. Let's imagine this is permissive and this is also permissive, this is non-permissive and this is also non-permissive. So let's imagine like this. If that protein, so let's say this is a B protein which is coming for polymerization action. Now in this permissive end it's fine. So when concerned to the permissive end, so they will allow this B protein to pass over here. But These protein, let's so they reached up to here. So let's say this is end, non-permissive end of, and next test protein. So this B protein or this B complex cannot pass through this point because this is a non-permissive end. And this, when this B protein reaches non-permissive end of and next test protein, this B protein will get fall off, or that will get dissociate once they meet non-permissive end of the test protein. So this is the idea of tester complex mediated termination. So when this B protein will meet non-permissive end of tester complex, as the result of this tester complex, the B protein will dissociate or the fall off. So this is the idea. And the same way this RB1, their B protein can pass through this region, but they cannot pass through the second tester complex because they have got the non-permissive end. So when they meet each other, this B protein will get fall off or they'll get dissociate. So this is the mechanism of, this is the basic idea of the termination. Now I want to discuss how this process actually takes place, and when what is the factor which influences the termination of this B complex. So better I'll erase here. So I just drawn a linear form of the tester complex. So I have to mention here. So this is TER site, let's say TER A, and this is TER protein. So this is TER protein. And what is the mechanism actually here? So what is the mechanism actually that takes place here? So they have got a non-permissive end. So this B protein, actually this B complex is going to react with, or it's going to meet a non-permissive end of DNA. Now, so they have got some sort of base which is present in between this B protein as well as the tester complex. Now, so what usually they do, so they will flip one of their bases apart. So that is the idea. So they will flip one of their base apart so that this B protein cannot read this frame. So B protein cannot read this frame. As the result of this flipping of the base, this B protein will get fall off or the B protein will get dissociate. So this is the idea of the termination in prokaryotes. So the B protein, what it is, the B protein, once they are going to meet this non-permissive end. So what the tester complex does, the tester complex will flip up one of their base apart so that the B protein cannot identify or they cannot read this frame. As the result, the B protein will get dissociate or the fall off. So this is the basic idea of the termination, and this is more than enough for understanding the termination process. And I hope you like the video. If you like the video, please hit the like button and don't forget to subscribe my channel.